Assalamu alaikum, my dear secondary 2 students. Our lesson today is about sensitivity or sensation. What is the meaning of sensation? Sensation is one of the functions shown by living organisms to maintain its life, where we find that sensitivity in plants is less obvious and sensitivity in animals more obvious. Sensitivity in human reaches its highest efficiency and accuracy. Sensitivity is the suitable response of living organism to internal or external stimuli to maintain its life. So, sensitivity means that must be stimuli and must be a response to maintain its life. Sensitivity or sensation is a vital process. Sensitivity in plants includes in response to touch and darkness in mimosa plants and tropism. Response to touch and darkness in mimosa plants. Okay, the morphology of plants is a primary, bulvinous, and primary raches or rachis, British or American, and the primary rachis ends with 4 secondary rachis, and each rachis ends with 2 rows of leaflets. So, bulvinous and primary rachis and 4 secondary rachis. Each one of the secondary rachis ends with 2 rows of leaflets. So, the morphology of mimosa plants leaves is mimosa is a plant, is mean in Latin shea plant. Leaves are composed of compound and pinnate. Compound and pinnate. Compound composed of many leaflets. Each has primary rachis which carries at its end 4 secondary rachis. Okay, had primary rachis and 4 secondary rachis. Each secondary rachis carry 2 rows of leaflets. 2 rows of leaflets. At the base of each primary and secondary and the leaflets also is a swollen structure called bulvinous or bulvinous. A response of mimosa leaflets to touch. If you touch a mimosa leaflets, its petiole soon drops as if it has wilted. Other neighboring leaflets soon will follow till the effect is seen in all leaflets. A response of mimosa leaflets to darkness. In day, the leaflets are held in a horizontal position, undergo wake movement, and at the night the leaflets hang downwards and fold their upper surface above each other, undergo a sleep movement. If you touch the leaflets, it will fold above each other, means a response to touch. The explanation of response of mimosa to touch and darkness. The swollen structure of bulvinous acts as a joint, like your joint, in this movement as follows. The cell walls of the lower half of the bulvinous are more sensitive than those of the upper half, and they play the main role in the movement. Okay, this is the bulvinous. The lower half of the lower part is this, and the upper part is this. So, when the plant is touched or the darkness, the lower surface of bulvinous shrinks. This leads to the water diffusion to the neighboring tissue and hence the leaflets drops. But when the stimulus is removed, the cell regain their rigidity and leaflets open once again. Okay. Tropism. Tropism is a Latin word, means a turning. Tropism movement represent the most common type of sensation in plant. Tropism is meaning a turning. It's the curvature of stem or root. The stem or root of plant when its sides are subjected to the factor stimuli as light, humidity, and gravity in unequal form, meaning only in one side, from one side, subject to the plant stem or root from one side. Types of tropism. Tropism classified according to the type of stimuli, determined according to the affecting factor light. Phototropism, the stimuli here is light. Geotropism, the stimuli here is gravity. Hydrotropism, the stimuli here is water. Phototropism. It is the response of plant parts of external stimuli, which is the light causing the curvature of the plants towards or away from the light. Phototropism, the external stimuli. To prove the occurrence of phototropism, first one, place trace settling on a cork disc floating on the surface of the beaker. The beaker half filled with water. Put the beaker in light-proof box at one side. Leave a small circular hole to admit light. Leave it for a few days. The observation is the stem inclines towards the source of light, so the root goes away from it. Conclusion is the stem is a positive phototropic and the root is a negative phototropic. The explanation. Curvature is due to the unequal growth of two sides in both the root and stem, where the side of stem which is away from the light grows more rapidly. So the stem curvature towards the light. The side of root is near to the light grows more rapidly, so the root curvature is away from light. Experiments to explain the phototropism. Bison-Jensen is a scientist. Explained these phenomena during his observation and the result of experiment on the old calyptile. Calyptile is the apical meristeme. Apical meristeme present at the stem and the root. Oat اللي هو شوفان. Steps. Calyptile is subjected to the light from one side. Okay. And cut off the tip, 1 or 2 millimeter of the tip, then it's subjected to the light from one side. The decapitated tip is returned or fixed again to a calyptile with gelatin. The tip is separated again from calyptile with a mica. Okay, observation. First one, the curvature of the calyptile towards the source of light. Okay, the stem is a positive phototropic. Plant loses its ability to bend towards the light. Third state is the ability to bend towards the light is restored, meaning that the plant bends towards the light. Fourth, no curvature. When the scientist is subjected a mica sheet, the conclusion is the tip of calyptile synthesizes a chemical substance called auxins that affected the growth and caused the tropism towards the light. The growth is due to the unequal concentration of auxin, leading to unequal concentration or unequal elongation. So the curvature takes place. The tip is the source of auxin, which causes the tropism. Auxin diffuse through the gelatin and affected the growth. Auxin can't diffuse through the mica. The explanation. The curvature towards the light, phototropism, is the result from unequal concentration of auxin on the two sides of calyptile, which causes the unequal growth of two sides. Auxins, they are a chemical compound secreted from the calyptile tip of plant and affected by the external factors. From most common example of auxin is IAA, indole acetic acid. Vent experiment. He made his experiment to prove the result of Bison-Jensen. As a step, he exposed an calyptile to an unidirection from one side, illumination. Then he cut off the tip and placed it on two blocks of agar separated by a metallic sheet like mica. He measured the concentration of auxin in each block, and the observation is 65% of auxin are accumulated in the agar block of the side away from the light. 35% of auxin are accumulated in the agar block side facing the light. Okay, second step, he put the blocks of agar on the decapitated calyptile, which is not subjected to the light. The observation is curvature of the calyptile tips. Okay, conclusion, auxin move from the side facing the light to the far dark side. This means that oxen hate light, go away from it. Curvature of the tip due to the difference in concentration of oxen in the added blocks. والسلام عليكم ورحمة الله وبركاته.